नमस्कार 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 सत श्री अकाल आदाब सलाम वालेकुम जय श्री राम. How are you guys doing? I hope you guys are well and your studies are going absolutely like a rocket. Okay, so my dear friends, my dear viewers, my dear extended family, I welcome you all to my YouTube channel PAWAR'S Bioscience Hub. You know my name. My name is Jayesh Pawar. And which chapter we are studying? We are studying the chapter Animal Kingdom. And today is the third lecture of Animal Kingdom. Okay. Today is the third lecture of Animal Kingdom. And what are we going to study? Today, I'm just going to teach you one single topic, Porifera. Okay. Only Porifera we are going to learn today. Okay. Before starting with today's class, let's quickly revise what we have studied in the previous class. Okay, we'll be very quick. Within a time period of approximately 9 minutes, 9 to 10 minutes, we will cover the revision. Okay, and after that, we will quickly move on to the phylum Porifera. There are no questions today. Okay. From tomorrow or day after tomorrow, you will be getting questions also. And yes, I told you that you will be getting the PPT. Okay, you will be getting the PDF. Now you will get it. Okay, from now on, you are going to get it. So now, my dear friends, let's start. What did we study in the previous lecture number 2? We studied about symmetry. Okay. The symmetry is divided into 3 types. Okay, just a minute. Yeah. Symmetry is divided into 3 types. Asymmetry, bilateral symmetry and radial symmetry. In asymmetry, even after passing a plane, we do not get, even after passing a plane through any of the organism, we do not get a single type of equal half. Okay, so that is called asymmetry. And what are the examples of asymmetry? The example is very simple, which we are going to learn today is sponges. Okay. After that comes radial symmetry. Now, what is in this radial symmetry? In radial symmetry, when we pass a plane, in radial symmetry, when we pass a plane, we get equal halves. Okay. But neither your NCERT nor your any of the preference book, and even I haven't mentioned how many symmetric we get in radial symmetry. Okay. After this, bilateral symmetry. Okay. So now, what are the examples of radial symmetry? The examples of radial symmetry are some sponges, Coelenterata and Ctenophora. Correct? Yeah. The example is some sponges, Coelenterata and Cnidaria also. Coelenterata and Ctenophora. I'm sorry. After this, moving on to the next type of symmetry, which is called the bilateral symmetry. Now, hearing the word bilateral, what do you understand? Bi means 2. Means even after passing up, when we pass a plane through any organism, in which we get 2 equal halves are called bilateral symmetry. It's called bilateral symmetry. Okay. Now, what is the example? The example is some invertebrates and all the vertebrates. Understood? Okay. After this, we are going to see we saw the body organization in the last class. Okay, diploblastic and triploblastic organization. So these diploblastic and triploblastic organization are germ layers. Okay, see, in diploblastic, there are only 2 germ layers, ectoderm and endoderm. Okay, but in triploblastic organization, between these 2 germ layers, there is one more layer. There is one more mesoglial layer called mesoderm, which is present in triploblastic organization. Now, when is the formation of germ layer confirmed? The formation of germ layer, whether the organism is going to be diploblastic or triploblastic is confirmed while it is in its while the organism is in its embryonic stage. Okay, understood? Correct. So now, what are the examples of diploblastic organization? The examples of diploblastic organizations are called are coelantrates. Okay, in coelantrates we see diploblastic organization, and from platyhelminths to chordates we see triploblastic organization. Understanding? Okay. After this we saw coelom. Now what is a coelom? Coelom is a type of body cavity formed by the mesodermal lining. Coelom is a type of body cavity formed by the mesodermal lining. Formed by splitting of mesodermal lining. I'm sorry. Okay. So now the coelom is also divided into two parts. Into three parts. Sorry. Coelomate, acoelomate, and pseudocoelomate. Coelomates are the animals who have the mesodermal lining present. Okay. Coelomates are the animals who have the mesodermal lining present completely. Acoelomates are the animals who do not have the mesodermal lining. And pseudocoelomates are the organisms in which the mesodermal lining is partially present and partially absent. Okay. Means it's not completely absent also and it's not completely present also. Now what are the examples? The examples are platyhelminths, okay, of coelantrates, sorry, of acoelomates. The example is platyhelminths. Coelomates have an example of annelids, arthropods, mollusks, echinoderms, okay, hemichordates to chordates. These are the examples of coelomates. Okay. Now and last but not the least, acoelomate. Sorry, pseudocoelomate. I'm sorry. I'm really sorry. Pseudocoelomate. So as I told you, in pseudocoelomate the mesodermal lining is neither completely absent nor completely present. It's partial. So these the organisms who possesses is pseudocoelomates who possesses a pseudocoelom organization are called Ashkelmans. Okay. After this we saw segmentation. After this what did we see? We saw segmentation. Now the segmentations are of two types. Okay. External segmentation and internal segmentation. In external segmentation, we looking at the organism, we can differentiate between its body parts. Okay. Take a simple example of cockroach. When you see the cockroach at night or whenever you see a cockroach, you can, if you have caught it and you have observed it clearly, okay, you have observed it carefully, you will get to see segments, means head, thorax, and abdomen. Okay. And in internal segmentation, okay, in internal segmentation the example is earthworms. Okay, earthworms show metameric segmentation. Understood? Earthworms show metameric segmentation. Okay, after this, the last but not the least topic of the second lecture is what is notochord. Okay, now what is a notochord? A notochord is a rod-like structure, mesodermally derived. Okay, and it is present on the dorsal side. Okay, it is present on the dorsal side. Animals who possesses notochord are from animals who possesses notochord are called chordates. Example is some annelids. Okay, I'm sorry. Yeah, sorry. I'm really sorry. What I said that the we'll just go a little bit back. I'm again telling you what is a notochord. Notochord is a rod-like structure present dorsally, which is mesodermally. Okay, and the animals who do not have this rod or this notochord are called non-chordates. Only the examples of non-chordates I am telling you: Porifera to echinoderms. Okay, from Porifera to echinoderms, the animals are non-chordates. After this, all the animals are chordates. Okay, so now with this, we have completed our revision within 11 minutes. Okay, within 11 minutes. Now we are going to learn a short topic, but very important. We are going to learn Porifera. Let's begin. So now, my dear friends, what to understand by the word Porifera? Porifera means pore bearing. Okay, Porifera means pore bearing animals, and these are the most primitive multicellular organisms. Okay, their habitat is mostly marine. Okay, their habitat is their habitat is mostly marine. Now, my dear friends, the criteria which we learned before this class, okay, we are going to try fitting each phylum in that criteria, means levels of organization, okay, segmentation, notochord, body organization, coelom, all these criterias. Okay, so now first of all, tell me what is the level of organization seen in phylum Porifera. In phylum Porifera, the level of organization is called cellular level. Okay, now after this, which type of symmetry is seen? The symmetry is asymmetrical symmetry. Okay, body organization is called diploblastic. Okay? These are the diploblastic organisms. You can see here this red part and this red part endoderm, white part ectoderm. Okay, in these organisms segmentation is absent, notochord is also absent. Now see here, these organisms are hermaphrodites. Okay, means male and females are present in same organisms, and the process of fertilization is internal. Okay, understood. Now, and the larval stage of sponges are free swimming, whereas the adults are sessile. So see here, I've mentioned here that, see, where the larval stage of sponges is free swimming. Okay, free means it freely swims everywhere. But when it enters its adult stage, it becomes sessile. Okay, so now what is sessile? Sessile means it gets permanently attached to a hard surface. Okay, it gets permanently attached to a hard surface. This hard surface can be rock or any other thing. Okay, now what to understand by digestion is intracellular. Yeah, the digestion means the digestion is performed within the body. Okay, now if someone is unable to understand what is intracellular digestion, just uh, imagine it as your home kitchen. Okay, just imagine it as your home kitchen. Your father, okay, your father brings vegetables. Okay, your father brings vegetables. Okay, and your mother is in the home, and your father brings the vegetables and gives it to your mother. Okay, just give me a minute. Now once more I'm going to tell you what is intracellular digestion. Okay, so see here, imagine it as your kitchen. Imagine it as your kitchen, as your home's kitchen. Okay, your father brings vegetable and gives it to your mother. Okay, now in the now what does your mother do? Your mother takes the vegetable from your father, breaks it into small pieces. Okay, add some spices, add some, what we say, adds oil, add spices, add the garlic. Okay, and after cooking it, gives it to you. Okay, and after cooking it, the you all, means your whole family, eats it. Okay, so now try to imagine. See, the sponges are there. Okay, the food comes from outside. Okay, the food enters the, what we say, the food enters the cell of sponges. I'm sorry, there was a big gap, but the food enters the food enters into the cell. Okay, the food enters into the cell. After that, the food is uh, taken by enzymes. Now, what are these enzymes? These enzymes are the cooks. Okay, which comes from another lysosome, which are in the lysosome. But after the organism captures the food, the enzymes comes from the lysosome. Okay, now what does these enzymes do? These enzymes break the food into small pieces. Okay, these enzymes break the food into small pieces, and after that, it is given to the whole organism. Okay, okay. So, did you understand what did I say? Do you want me to explain it once again? So, see, the food enters the cell. Imagine again. I'll tell you. Imagine it as your kitchen. Your okay, your mother as the enzyme. Okay, your mother is the cook. Okay, your mother is sitting in the another room. Okay, let's name it as lysosome. After the food is brought by your father. Okay, after the food is brought by your father or vegetable vendor or anybody. Okay, the enzymes or the cook, means your mother, comes out from the room, comes out from the another. Okay, and what is this room called? This room is called lysosome. Okay, lysosome or lysosome. Okay, lysosomes. Okay, after coming out from the lysosomes, your mother receives the receives the food from the vegetable vendor or anybody or the delivery guy. Okay, your mother cooks it, and after that, you are given the food. It's as simple as that. Okay, understood. Now the next part. Now the very much important part, which is water canal system. Okay, now what is this water canal system important for? So see here, this water canal system is important for gathering food, respiratory exchange, and excretion. Okay, now first of all, I'll read this out, and after this I'll show you the video. Okay, so what is the mechanism of water canal system? The water enters in the body of sponges through the pores ostia, through the pores which are called ostia present on the body wall. Okay, after this, water gets into the hollow cavity which is called spongiocele. Okay, and after the whole thing is done, water is excreted out through the osculum. If somebody is not understanding, I'll tell you. So, have you eaten the coconut? The small one, not that big green color. Have you eaten a coconut? The outer body of the coconut is hard. Okay, the outer body of the coconut is hard. Okay, imagine it as the body wall of sponge. Okay, now on that body wall there are pores present. There are pores present on the hard outer surface of the coconut. Imagine those pores as ostia. Now what will happen? The water will get into the coconut with the help of ostia, from the ostia, sorry. Okay, and after getting in, the water will get stored in a hollow cavity called spongiocele. Now imagine as the white part which we eat. Okay, which we eat, imagine it. Okay, it is soft. So imagine it as spongiocele. The water is stored in it. After this, water is excreted out through osculum. The two eyes present on the what we say present on the coconut. So after this, after using the water, after doing all these things, okay, gathering food, respiratory exchange, and excretion, the water is excreted out through those two openings called eyes of the coconut or osculum. Understood? Okay. After this, see here. Now, the food in the water won't be captured on its own. Okay. So for this, there are conocytes. There are conocytes. What are these conocytes? These conocytes are collar cells, which produces currents. Okay, which produces water currents and lets the organism capture the food. Understanding? Understood? Tell me if there is any issue. Okay, now see here. There are three types of canal system. Asconoid. You don't need to go deep into this. Okay, you don't need to go deep into this. Just remember the names and the complexity. Okay, first is Asconoid. Sorry, Asconoid. After it is Syconoid and Leuconoid. Okay, Asconoid, Syconoid, Leuconoid. From going Asconoid to Leuconoid, the complexity increases. Okay, the complexity will increase. Understood? Okay, so I hope you understood the complexity. Okay, now let's move on to the next part. See, just give me a minute. Yeah, okay, so now very short part. Okay, the body is formed by spicules and spongins. Okay, so now what is the spicules? Spicules is hard needle-like structure or star-shaped structure. Okay, hard needle-like structure or star-shaped structure made up of calcium carbonate or silica. It helps provide rigidity. Okay, it provides rigidity to the organism. Understanding? Okay, it provides rigidity to the organism. And what are spongins? Spongins are soft flexible proteins forming the elastic framework. Spongins are the soft elastic proteins forming the elastic framework of the elastic framework of sponges. Okay, so now see here, there are some, I've added the examples, I've added the pictures of sponges. Okay, so this is Sycon. Okay, this is Sycon. This is Spongilla, and this is called bath sponge. Okay, this is Euspongia. In simple words, in the common language, it is called Euspongia. Okay, in common language, it is called bath sponge, and in scientific language, it's called Euspongia. Completed? Okay. So now, my dear friends, let's quickly read up the NCRT. All the points are covered. Okay, all the points are covered in this NCRT. Okay, so now let's start reading. 4.2.1 Phylum Porifera. Members of this phylum are commonly known as sponges. Okay, they are generally marine, which we saw. They are mostly asymmetrical animals. See, I told you they are asymmetrical. These are primitive multicellular animals. Okay, and have cellular level of organization. Okay, sponges have a water transport or canal system. This also I told you water enters through a minute pores which are called ostia in the body wall into a central cavity called spongiocele, from where it goes out through the osculum. This is the pathway which I explained you using the coconut example. Okay, this pathway of transport is helpful for gathering food, respiratory exchange, and removal of waste. Okay. Choanocytes or collar cells line the spongiocele and the canal. Okay, wait a minute. Lines the spongiocele and the canals. Digestion is intracellular. This I told you. The body is supported by a skeleton made up of spicules and spongins. This also I told you what are spicules. Spicules are hard needle-like structures which are formed of silica and calcium carbonate and helps in providing rigidity. Spongins are soft fibers. Sorry, spongins are soft fibers which spongins are soft. Okay, so spongins are soft fibers providing the elastic framework. Okay, after this, these point also be covered that sexes are not separate, means the organism is hermaphrodite. Okay, means eggs and sperms are produced by the same individual. Sperm can reproduce sexually by fragmentation and sorry, where it is sponges reproduce asexually by fragmentation and sexually by formation of gametes. Okay, this line also we covered. Fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult. This line I explained you. Okay, so now with this, my dear friends, thank you for watching this lecture.